to the next video of different types of immunoglobulins. Here we'll be discussing immunoglobulin A, immunoglobulin D and immunoglobulin E. Now the first two subclasses, namely immunoglobulin G and immunoglobulin M, has already been discussed in my previous video. If you have not yet checked it out, please go and make sure that you check it out first and then come back to this video. Now starting with immunoglobulin A. Now as you can see, there we have two fundamental structural unit. One is this fundamental structural unit of an antibody and the other one is this fundamental structural unit of an antibody. Now both these fundamental unit of antibodies are stabilized together via a J chain. Now this J chain is the same J chain which we have analyzed during the discussion of immunoglobulin M structure. Apart from this J chain, we have got an additional component which is known as the secretory component. The appropriate diagram of the same will be pinned up at the end of the video. Now basically, coming to immunoglobulin A, it is a dimer which has got 10 to 15% concentration in the blood. Along with the J chain, it has got an additional secretory component which is also attached. Among all the classes, 2/3 production of the immunoglobulins is of immunoglobulin A. Now while reading this particular statement, don't you think that this particular statement contradicts with the first statement, where the third statement says that among all the classes that is produced in the blood, 2/3 production is of immunoglobulin A. Then how come its presence drops drastically in the blood? That is only about 10 to 15% is present in the blood, whereas its presence must be in much higher concentration. Then seeking the answer to this particular question, we need to move to the next statement. Now coming to the next statement, it says that immunoglobulin A perform transcytosis to reach the external secretion. By analyzing this particular part of the entire statement, one thing is very much clear, that immunoglobulin A does not have an active role within the blood circulation, and therefore it needs to reach an external secretion, or it needs to reach the external secretion where it might, or rather it have the active role which we are talking about. Now in order to understand this statement, you need to understand two things. One, what is transcytosis, via which the immunoglobulin reach to the external secretion, and what in the first place is external secretions. So basically external secretions are the secretions like tears, saliva, mucus, that is mucus which is present for example in the respiratory tract, in the gastrointestinal system and so on. Now since we have understood what external secretion is, now we need to understand the process via which it reaches the external secretion, that is transcytosis. Now before understanding transcytosis, I need to make it very much clear that we know about phagocytosis. Now what is phagocytosis? It is when a cell engulfs some external thing within itself. The process is known as phagocytosis. And when a particular cell, it wants to secrete something, it wants to speed out something from its inner environment to the outside environment, the process via which it does it, it's known as exocytosis. And when both the process, that is phagocytosis and exocytosis, takes place simultaneously, it is known as transcytosis. Now since we have understood what transcytosis is, then we need to also understand that how does it hold true for immunoglobulin A transport. Okay, then for this, let's come to this particular schematic diagram. In this diagram, I have drawn, or I have shown a schematic picture of a particular or a peculiar blood vessel rather. Now in this blood vessel, I have drawn some endothelial cell on its surface. Now see, there are endothelial cells on both the sides, but just for illustrative purposes, I have drawn the endothelial cells in one side only. Now here there is the immunoglobulin A which is synthesized in the blood via the B cells. Now this immunoglobulin, since it has to go to the external secretion, it needs some path to go. Now see, immunoglobulin A cannot be transported directly from the blood vessel to the external environment. So for that we need some medium for its transportation. So one of the medium is receptor. That is, via interacting with the receptor, it can move out of the blood vessel and reach its destination, that is to the external secretion. Now see, here now the presence of J chain helps the immunoglobulin A to interact with the surface receptor present in the endothelial cell surface. Okay, once the interaction is established between the immunoglobulin A and the surface cell receptor or the cell surface receptor of the endothelial cell, then phagocytosis is performed. That is, this endothelial cell will engulf the entire immunoglobulin along with the receptor within itself, okay, in a phagocytic vesicle, as you can see here. Now this phagocytic vesicle needs to move out of the endothelial cell, so therefore the endothelial cell will perform exocytosis to let it go outside the cell along with the receptor being intact with the immunoglobulin A, and thereby, see here you can see that the immunoglobulin A attached with its receptor via which it entered the endothelial cell is packed in a phagocytic vesicle, rather an exocytic vesicle. That is, first step, the immunoglobulin A will interact with the surface receptor, then it will perform phagocytosis. Via phagocytosis it will move within the endothelial cell, but it needs to move out, so therefore this endothelial cell will perform exocytosis and will transport the immunoglobulin A along with the receptor via which it interacted at the initial stage to the outer environment in a protective vesicle. Now this is how immunoglobulin A gets transported from the blood to the external secretion so that it can defend incoming pathogens, right? So this is transcytosis, and this is why the concentration of immunoglobulin A drops down to 10 to 15% only, whereas 2/3 of the production is of immunoglobulin A, because immunoglobulin A needs to move to the external secretion from the blood and therefore its concentration drops. So immunoglobulin A performs transcytosis to reach the external secretion, and during the transcytosis the secretory component is added that was actually a part of the receptor. Now as an extra information, now this secretory component has also got some role. Simply it doesn't get attached to the immunoglobulin A. Basically, now whenever this immunoglobulin A will be transported to the external secretion, then it may encounter some harsh digestive enzymes, and those harsh digestive enzymes might cause damage in the hinge region. See, this is the hinge region, right? In these regions of the immunoglobulin A, and it is quite possible that it may damage the entire immunoglobulin. So for its protection, this secretory component is added, which helps protecting the antibody from any harsh digestive enzymes. So that was an extra information for you. Now this immunoglobulin A has got two subclasses, that is immunoglobulin A1 and immunoglobulin A2. These are two isotopic form of immunoglobulin A. Now it has got predominant role in infections like influenza, polio, etc. Yes, definitely. So those infections that occur via the respiratory tract, for example influenza, will have to encounter its protective army, that is immunoglobulin A. So therefore, as I have said, that immunoglobulin A has got a predominant role in external secretion. So therefore those infections that occur via respiratory tract, GIT, okay, those infections would have to face the immunoglobulin A, and the immunoglobulin A on the other hand will defend us from such kind of viruses that enters in our body via the external secretion. So that's all for immunoglobulin A. Then comes to immunoglobulin D. So basically it has got a monomeric structure, for example a typical antibody structure, and it is present as a receptor on B cells. So B cells are the mother of all the immunoglobulins. As I have already discussed, that immunoglobulins are a product of exclusively the B cells, and immunoglobulin D is present only as a receptor on B cells. However, the specific role is unknown. Then coming to immunoglobulin E, it's again monomeric and plays an important role against the allergens. That is, what are allergens basically? So allergens are anything and everything that may cause immediate reaction. Alright. Then comes immunoglobulin E receptors are present on basophil, mast cells and eosinophil. Yes, the reason it's very logical, because immunoglobulins can only recognize the pathogen. For its neutralization, we need some specialized professional cells, and these professional cells will be basophils, mast cells and eosinophils. Basophils, mast cells and eosinophils have got receptor for immunoglobulin E. After recognition of the specific allergens, immunoglobulin E along with the allergen will come for help to this basophil, mast cells and eosinophil via the receptor which is present within the on the surface of the basophil, mast cells and eosinophil, and thereafter the neutralization activity will be accomplished. So that's all for the immunoglobulin structures. Hope you have understood it well, and in our next session we will be discussing about the immune cells. And till then, if you have liked the content in this video, please do subscribe, share and like this video. And also if you have got any other subject recommendation, you can drop your recommendation in the comment box below. Thank you very much.